Welcome to What is SDH, How is it related to cancer, and what can be done about it? Please remember the information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Sarah Rothschild and I am your moderator for today. Before I begin to introduce our speaker, I would like to thank SDHRA, GSI, and Just Support UK for helping to get the word out about this seminar and working closely to spread the word. I would like to introduce now our presenter today, Dr. Eyal Gottlieb. Dr. Gottlieb received his BSc in agriculture from the Hebrew University and an MSc and a PhD in molecular cell biology from the Weizmann Institute of Science. In 1998, Dr. Gottlieb moved as an EMBO fellow to the University of Chicago and later became a Leukemia and Lymphoma Society Special Fellow at the University of Pennsylvania. In 2003, he moved to the Cancer Research UK Beatson Institute in Glasgow and was appointed a professor of molecular cell biology at the University of Glasgow in 2009. Since 2016, Dr. Gottlieb is the Laura and Isaac Perlmutter Chair of Cancer Research, Faculty of Medicine, Technion-Israel Institute of Technology in Haifa, Israel. Dr. Gottlieb studies the metabolic adaptations that support tumor growth under metabolic stress. Specifically, he explores vulnerabilities induced by the loss of the metabolic tumor suppressors fumarate hydratase and succinate dehydrogenase. Please note that he is not a clinician. He does not see patients. He is a scientist. So any questions that you may ask, just keep that in mind. Dr. Gottlieb, you may begin your presentation now.
Hello, everyone. My name is Eyal Gottlieb. I work at the Technion, the Israel Institute of Technology in Haifa, Israel. And my research focused on cancer and metabolism in general. In particular, I'm working on succinate dehydrogenase mutations in cancer. I do want to disclose that I'm also a founder of a company named Metabolism that is engaged with identifying vulnerabilities in cancer metabolism and trying to develop small molecule compounds as potential drugs to treat those diseases. Before we go deeper into succinate dehydrogenase and cancer, I'd like to set up some understanding about language and tools that I'm going to discuss. So what are we made of? We are an organism that is made of basically like any other living creature or any other item in this world. We're made of atoms that are assembled together into molecules. In this part you see several atoms, carbon, hydrogen, oxygen, assembled into a DNA stretch, which is a large molecule that consists of billions of such atoms. Molecule combined together makes a cell, a living cell. That's the smallest living part in our body. Cells are then connected together, cells of the same type, to make a tissue. A tissue will have a particular function, and it's made of relatively similar cells. And then multiple types of tissues will make a functional organ. It can be a liver, a heart, a brain. It's made of multiple tissues, which are made of multiple cells. And then several organs will generate a system, which can be, for instance, the digestive system, or a brain is a system on its own, etc. And then all those systems will make us, or will make an organism in general. It doesn't have to be a human. So this is just the language. The scale is totally incorrect. I mean, atoms are much, much, much smaller than are presented here based on the scale of a human. But this is just an indication for language purposes, to understand each other. One important thing that I like to talk about at the moment is called the central dogma of molecular biology. This is basically how living cells are organized and function. What we have is a template, a genetic template called DNA. It's a double helix molecule. It's a very large molecule coiled together in the nucleus of the cell. And it generates, it encodes for all the ability to make proteins. So along this DNA stretch, there are genes. A gene is a part of a DNA that can be used as a code to make a protein. But the way it makes protein is that a gene is transcribed into an RNA stretch, and it's called messenger RNA in this case, or mRNA, which is a smaller type of molecule, not too dissimilar chemically from DNA, but it's much smaller and it can move around, and there are multiple copies of every gene can be transcribed to hundreds and thousands of mRNA molecules. These mRNAs are then being translated into protein in a process, in an organelle called ribosome. So the RNA is the template to make proteins. And this is how genes control the cellular function by making codes for making proteins. So this is the central dogma of molecular biology in our cell. And in relation to cancer, along this pathway, many things can go wrong, and that can lead to cancer formation. For instance, mostly the DNA can be altered. It can be a mutation, which is a mistake in the structure or the stretch of the molecules that makes the DNA. These mutations will lead to dysfunctional protein eventually. So these are called mutations. Also, they can be deletions. Part of the DNA can be removed and then cannot be transcribed or translated into proteins. It can be amplified. It can be more than the normal 2 copies per gene that we have in our cells. And then you make more RNA and more proteins. And that can also potentially cause diseases such as cancer. And in some cases, and it's relevant as well to SDH-dependent cancer, it can be silenced. There can be a chemical modification on a gene, such as methylation. It's adding a small decorated molecule called methyl that shuts down the transcription process. So if a region is methylated, typically it's not transcribed and therefore does not translate into proteins. So these are the modifications that can occur on the DNA level. And they will lead to, this mutation will be then transcribed into the RNA and translated into protein. If we get overactivity of a protein eventually, we call the gene that leads to cancer, we call the gene that generated an oncogene. And if we get underactivity or lack of activity, lack of function of the protein due to deletion, mutations that didn't enable proper function of the protein, or methylation, as I mentioned, that prevent the transcription of this gene, and the loss of the activity leads to cancer formation, this gene is called tumor suppressor gene. In the case of succinate dehydrogenase, all succinate dehydrogenase related genes are indeed tumor suppressor genes. So let's talk a little bit about metabolism. Metabolism is really all the processes that happens in our cells that leads to survival, to generate energy and to grow, basically, to make biomass. So even in a planet like this, what we have is a lot of metabolism going on in order to sustain survival. Now, particularly if this little plant needs to grow into a tree, you can imagine how much metabolic processes, how many metabolic processes are required in order to grow into such a big organism. So building up biomass and maintaining the energy to support it is what metabolism is all about. But it is also related to genetics. As you can imagine, if we have a genetic blueprint to make an elephant, the size of an elephant will be very different than the size of a snake, for instance, down here, or a frog. So the genome, the part of our DNA that codes for what we are, also dictate to some extent, our size. So metabolism is linked also to our genetic information. But not only. It also relates to environmental effects, particularly if we get fat due to inappropriate eating. We grow. We put different tissues, particularly fat tissues, and we gain weight. This can be partly related to our genome. Some people are more inclined to get fat and some less. So, but by and large, this is affected by how much we consume and how much energy we waste, basically. What does it have to do with cancer, all this? Well, cancer is a process of growth. What we see is a small few cell tumor, small tumor, that then keep on growing until they reach a larger stage. Now, I want to emphasize that the growth of a tumor is typically not by growing of the cells themselves, but actually by making more and more cells uncontrollably. But nevertheless, for a cell to continue to divide, it's a metabolic process. It needs to double its biomass. So what is a cell made of? So this is just a schematic illustration of a cell. What we have is the nucleus where the DNA is. We have multiple membranes made of fatty acids and different fat molecules. We have lots of proteins floating around, which we do not see here. And then there are organelles. The nucleus is one such organelle. But one I'd like to emphasize here is the mitochondria, these structures here. These are organelles that are very important in maintaining the energy of our cells and also in providing precursors for large molecule that makes our biomass, for proteins, for nucleotides, DNA, RNA, and for lipids. So these are the mitochondria. In fact, the dehydrogenase is a complex of proteins that sits inside the mitochondria, and I'll talk about it soon. So you can see that to make a cell to multiply, we need to build all these components, and that's a big metabolic problem for the cell. This is how it looks. Again, it's an illustration, but it's a very dense structure of cells. And by and large, the dry biomass of the cell consists of proteins, lipids, and nucleic acids, which are DNA and RNA. So to make biomass, also the cells need to assemble small molecules or precursors into larger molecules. For instance, in the case of proteins, there are about 20 types of amino acids that a stretch of them will make a protein. It can be a stretch of a few tens of amino acids up to a few hundreds or even a few thousand of amino acids that will make a protein. And this folded stretch of amino acids will then have a function. And there are a few tens of thousands of different proteins in our cells. This is just the buildup of proteins. But as I mentioned, DNA and RNA are also macromolecules. They are made of smaller molecules called nucleotides, mentioned here. But also lipids are made of fatty acids, for instance, and some other smaller parts as well. So the bottom line is that to grow and to develop biomass, a cell needs to assemble small molecules into larger molecules that contribute to the biomass. And all this basically is a big metabolic network. In this illustration, I'm going to talk about particularly the contribution of glucose. Glucose is sugar that we consume, and actually is a very important source of energy, but also an important source of multiple biomass contributing elements. So glucose can be converted into another molecule called pyruvate in the part of the cells called the cytoplasm, and then this is a process called glycolysis. It can make energy. ATP is a molecule that stores energy, and it's mentioned here. But this pyruvate makes more energy if it gets into the mitochondria, and this is just an illustration of the mitochondria, where it gets further oxidized or break down into carbon dioxide and water in a process called the Krebs cycle or the TCA cycle. The Krebs cycle enable further energy generating functions, which I'm not going to mention here, but they are very important to really converting all the glucose molecule into water and carbon dioxide, and to make a lot of ATP from it. So therefore, the TCA cycle or the Krebs cycle is very important for energy production. But beyond that, it's also an important precursor for many molecules that are essential for biomass. For instance, all these in brown are amino acids that are derived by and large from the Krebs cycle or from glycolytic intermediates. And they are essential for building up proteins. Also, nucleotides that are required to build DNA and RNA derived from these amino acids from glycine, aspartate, from glutamine, and they make these nucleotides together with a part from the glucose, which is called glucose 6-phosphate, which is converted to another sugar moiety called ribose 5-phosphate. Altogether, that makes the DNA and the RNA. And then also the fats are made of fatty acids derived from another molecule that come from the Krebs cycle called citrate or citric acid. So altogether, the citrates contribute to fatty acid biosynthesis and then to lipid biosynthesis. So what we can see here, how essential glucose is, not just for energy, but also for biomass. And with that also, we see how essential the TCA cycle, the Krebs cycle, for maintaining cell survival, both by energy and by building up the biomass. The reason I'm emphasizing it because succinate dehydrogenase sits right here in the Krebs cycle. It's an enzyme, it's a protein complex that is part of the Krebs cycle. And its this functionality leads to defects in all these processes that I just mentioned. Historically speaking, the field of cancer and metabolism really initiated almost 100 years ago with early discoveries by a German scientist called Otto Warburg, who also very much discovered all the energy producing processes in the mitochondria. Later on, he suggested that cancer cells have defects in those energy processes in the mitochondria, and therefore they produce a lot of lactic acid due to lack of full conversion of glucose to carbon dioxide and water. As said, he claimed that all cancer should be derived from defects in the mitochondria. Today we know that this is not the case. The majority of tumors or cancers have functional Krebs cycle and mitochondria. It's only those rare types of tumors, such as succinate dehydrogenase deficient tumors, that do not have functional mitochondria. But nevertheless, he initiated the idea of exploring metabolic changes that occur in the process of cancer formation. There are several other important landmarks that were discovered over the years. For instance, the understanding that tumors have regions which oxygen is limited due to lack of availability of blood supply to these tumors that deliver oxygen as much as other nutrients as well. The understanding that they consume a lot of glucose, something that is utilized in the clinic by labeling glucose radioactively and imaging it with a machine called positron emission tomography, or PET, and this is a process to vision tumors in patients. Further on, the understanding how cells respond to lack of oxygen, the discovery of a protein called hypoxia-inducible factor, which is basically it's a protein that binds to DNA and regulates the transcription of many genes that are associated with changes in metabolic processes due to lack of oxygen. So it's a master regulator of the metabolism, particularly under low oxygen conditions. The hypoxia is low oxygen conditions. So this discovery of HIF, or hypoxia-inducible factor, is a very important landmark. It was done in the 1990s. And maybe finally here, in relation to this webinar, the discovery in the year 2000 that succinate dehydrogenase, in this case subunit D, was firstly discovered as a tumor suppressor gene, meaning that its loss can lead in this particular study to hereditary syndrome called hereditary paraganglioma. Nowadays we know that succinate dehydrogenase loss of function, mutations and deletions, and methylation that shut down its transcription can occur in multiple types of cancer, still rare events, including GIST and renal cancer and paragangliomas and adrenal tumors such as pheochromocytomas. So as I mentioned before, succinate dehydrogenase, the complex, sits right here in the heart of the Krebs cycle inside the mitochondria. This is where the protein lies. There are 4 proteins that make succinate dehydrogenase. They're called SDH-A, B, C, and D. And each one of them, when mutated or deleted, or when we get a dysfunctional complex, would potentially cause cancer at a high percent. So it is quite surprising, actually, that a loss of function of such a key process, such as the Krebs cycle, is leading to cancer formation rather than to, say, cell death, which one would expect. But we know quite a lot about the structure of this complex. It's right here. This is the membrane, and this is succinate dehydrogenase A. This is B. C and D are embedded in the membrane. This is just an illustration of the molecular structure of this protein complex. This is its function. I'm not going to go into too many details, but as said, germline or somatic mutations meaning they are not in the germline, but then occur later in adult life, but also loss of function in these can cause cancer in some cases and in some tissues. So how does the loss of succinate dehydrogenase contribute to cancer formation? So this actually has to do with the discovery of the way hypoxia-inducible factor I mentioned before, HIF, is regulated normally. So typically this is a protein that is very unstable. It's being produced and degraded continuously in our cells in the presence of oxygen. So this is molecular oxygen. This is what we breathe. It's made of 2 atoms of oxygen atoms. Now, there is an enzyme called PHD, or prolyl hydroxylase, that splits those 2 atoms of oxygen into 2 separate atoms rather than the molecule, and one of them is decorating this HIF on a particular place called amino acid called proline. Therefore, it's called proline hydroxylase. This OH is an hydroxyl. This enables a binding of a different molecule called VHL that actually puts ubiquitin, that's a small stretch of amino acids, on HIF, and that mark it for degradation. So it sends it to the garbage place of the cells, and therefore HIF is always made and degraded because it gets hydroxylated through this prolyl hydroxylase. So what does it have to do with SDH? Well, I mentioned only the fate of one atom of this oxygen, but the other one is also used in this reaction in order to convert a molecule called alpha-ketoglutarate into succinate. Okay, now we're getting nearer. Succinate is the same molecule that succinate dehydrogenase is using as a substrate for its activity, and here it's the product of this reaction. So what we and others demonstrated is that when succinate dehydrogenase is dysfunctional, there is a high accumulation of this molecule called succinate that then basically goes out of the mitochondria into the cytoplasm, and it inhibits the activity of prolyl hydroxylase. It prevents the conversion of alpha-ketoglutarate into making more succinate. At the same time, it prevents the hydroxylation, the adding of the OH group to the proline residue on HIF, and therefore HIF is no longer degraded and it's stabilized. So we get a response as if the cells are exposed to low oxygen, even though they are in the presence of oxygen. So they change the metabolism, and HIF has a supportive role for tumorigenesis. It does not explain all the reason why a cell becomes cancer, but it is partly explaining the process of cancer formation due to the loss of succinate dehydrogenase. Actually, the genetics here help clarifying this as well. This is a genetic, what is called oncoplot, where many genes are followed, and every column is a patient, where you see what type of genetic alteration one patient have, and there are about 100 patients in this study. Okay, and what you see, there are germline mutations. For instance, these are all paraganglioma patients. These are not GIST patients. But you see many SDH-B mutation in germline. You see SDH-D mutation in the germline. You see deletions, somatic deletions of succinate dehydrogenase B, SDH-B in many types of paragangliomas. Also, one can see mutations in the EGLN1 gene, which is the code for the protein called PHD, the same enzyme that put the hydroxyl group on the HIF. And also HIF itself is mutated in many patients with paraganglioma. And that is the mutation here is exactly on the site where the hydroxyl group is given. Therefore, these mutations made a stable HIF proteins. So the genetics, together with the biochemistry that I mentioned before, actually put a kind of a pathway that explain, or at least partly explain, the tumorigenic effect of the loss of succinate dehydrogenase, which is associated with the VHL function, with the prolyl hydroxylase function, and with HIF functions as well. Now we know much more. This was easy so far. What we know right now is that there are many other enzymes that use oxygen and split molecular oxygen. Collectively, they are called dioxygenases, and they all use alpha-ketoglutarate and make succinate. And apart from regulating HIF, they're also regulating the methyl groups that are put on DNA and on regulatory elements of the DNA called histones. These dioxygenases are so-called demethylases. They remove those methyl groups. So when they are inhibited, there is more methyl groups along the DNA stretches, the so-called hypermethylated phenotype. So what we know is that the loss of succinate dehydrogenase would lead to the accumulation of succinate that will inhibit those dioxygenases that regulate HIF, but also will lead to hypermethylation as well. There are some other metabolic genes that are either deficient or gain a new activity in cancers called fumarate hydratase or FH and isocitrate dehydrogenase, IDH, and collectively the loss of function of FH or the new function of IDH leads to the accumulation of other molecules such as fumarate and 2-hydroxyglutarate. Again, forgive me for these words. And they all can inhibit dioxygenase and have somewhat a similar effect as succinate in succinate dehydrogenase deficient tumors. So this is what we know basically about the mechanism of tumorigenesis following the loss of succinate dehydrogenase. Is there anything we can do about that? Or is there anything we can try and do in order to develop new drugs that will treat SDH-deficient cells? Now, here I'd like to show you how generally the concept of treating cancer goes. So there are several things. First, we have the old-fashioned, should we say, chemotherapy, which is a less specific toxic drug that is more toxic towards cancer cells than the normal cells in our body. For instance, it prevents DNA synthesis, and therefore cells that need to multiply are more affected than cells that do not need to be multiplied. But here I'd like to focus on the so-called targeted therapy, meaning that we know exactly what molecular targets the drug that we are making is attacking. In the case of oncogenes, those genes that are mutations or overexpression of them would lead to cancer, they generate a process called oncogene addiction. And therefore, we can try and design small molecules that will specifically block the function of these oncogenes. And that's a simple, straightforward, and reasonably successful approach to target oncogene-driven tumorigenesis. Once we turn the oncogene off, then the cells can stop proliferate or even die, and we get a tumor regression. However, when we have cancer forming due to lack of a tumor suppressor gene, this approach is not valid because you cannot try and inhibit something that is not there, like in this case succinate dehydrogenase. So what do we do then in order to design targeted therapy for tumor suppressor loss? Here comes to our help an approach called synthetic lethality. It's an approach of identifying what other genes becomes essential when the gene of interest, the tumor suppressor, is lost. So for instance, when succinate dehydrogenase is dysfunctional, is there any metabolic or anything else, any other gene that produce something that now it becomes essential? This something, or this protein that is produced by the other gene may not be an oncogene on its own. It will not, when it's active, generate tumors, but it will sustain the survival of tumors in which, in this case, succinate dehydrogenase is deficient. This is the process of synthetic lethality, or the so-called non-oncogene addiction. The cancer cells become addicted to another protein, even though it's not an oncogene. And this is what we're trying to look for in the case of tumor suppressor gene loss, and particularly in succinate dehydrogenase, because succinate dehydrogenase is such a key metabolic enzyme. Therefore, one can imagine that in order to survive the loss of succinate dehydrogenase, the cells must find a backup mechanism or circumvent this fundamental deficiency in the Krebs cycle. So if we will identify metabolic processes that enable the cells to survive, we can try and target them with small molecules and so-called drugs, with the hope that other cells, normal cells, will not be affected by this drug, only the SDH-deficient cells. So this is the process of synthetic lethality as an idea how to target SDH-deficient tumors. This is a real map of human metabolism. Every dot here is a metabolite. Every line is a process or an enzyme that converts one metabolite to another. Here in the middle is where SDH lies. Okay, so now it's a big question to try and identify what change in this landscape occur due to the lack of succinate dehydrogenase and what part of these changes is essential for SDH loss, but is not essential for cells that have functional succinate dehydrogenase. So you see why the search into it is actually quite complicated or quite time-consuming and requires a large-scale analysis of the metabolic landscape plus validation of every change and to understand whether it is essential on the one hand and whether it is toxic on the other. Toxic meaning that it will also, if we will inhibit it, it will also affect some other normal cells. So that's not a trivial approach, but this is the way to go around succinate dehydrogenase deficiency in particular. I'll give you a few examples for what may happen or where do we stand maybe today. So here is succinate dehydrogenase in this box. It's in the TCA cycle. It's also part of the energy producing process. I didn't spend too much time on that. So what us and others have already been demonstrated that it becomes essential in cells that are deficient in succinate dehydrogenase. For instance, this enzyme called pyruvate carboxylase, or PC, is a very important enzyme that transfer a molecule called pyruvate, the end product of the breakdown of glucose in the cytoplasm, into the Krebs cycle in a way that maintain the amount of Krebs cycle metabolites. So while there is still a break in the Krebs cycle due to the loss of SDH, you can still replenish some of the metabolites inside the Krebs cycle, which are essential for biomass processes, and to grow in order to develop and divide. So this pyruvate carboxylase might be a good target, though at the moment there are no small molecules that can inhibit it. And also we know that pyruvate carboxylase can be also quite an important enzyme in normal tissues, particularly in the liver and some other tissues as well. So there is a risk of toxicity if we will be successfully inhibiting this enzyme. So it may or may not be a good target, but at the moment we don't know for sure yet. Some other interesting possibilities are to do with the process of glycolysis, the breakdown of glucose. Since the Krebs cycle is defective and there is no proper energy-producing processes in the mitochondria, the cells by and large rely on the energy that is produced by the breakdown of the glucose in the cytoplasm. Under these conditions, the cells must get rid of pyruvate, which cannot be properly converted into carbon dioxide and water in the mitochondria, so it is secreted as lactate. Okay, there's an enzyme called lactate dehydrogenase, produce lactate, and then it gets out of the cells. So these steps, lactate dehydrogenase and the transporter of lactate called MCT, or monocarboxylase translocators, they are potentially good targets to block the process of energy production by glucose breakdown in the cytoplasm and not in the mitochondria. There is one molecule in the clinic here that is a MCT1 inhibitor. There are 4 of them. And actually it is in a phase 2 clinical trial for different types of cancers, because there are many types of cancers that require glycolysis, as partly Warburg demonstrated almost 100 years ago. And it potentially can be interesting to explore this site, which I said currently is in clinical investigation, particularly in the UK. It's a molecule made by AstraZeneca, and they made MCT1 and 2 inhibitor. Now they're making MCT4 inhibitor. I'm not sure where it stands in terms of how soon it will get into the clinic. How safe it is, we don't know about it. Another approach might be simply to block the uptake of glucose. We can block the transporter of glucose into the cells. So that's quite a risky approach if you think about it, because many normal tissues require glucose, heart, brain. So blocking this might be a bit risky. Nevertheless, if you can just cut it down a little bit by, I don't know, 25, 30, 50%, maybe the succinate dehydrogenase deficient cells will be more sensitive to that than other normal tissues. And in fact, there are several companies that already developed glucose transporter inhibitors, and only a couple of weeks ago, a poster in a cancer meeting on cancer metabolism demonstrated some efficacy in preclinical study of one of these inhibitors, particularly in succinate dehydrogenase deficient tumors. So this is totally new. I mean, only maybe last month it was presented. Another potential use is the inhibitor of another enzyme called glutaminase, or GLS. This glutaminase is also important to convert an amino acid called glutamine into a Krebs cycle metabolite called alpha-ketoglutarate. So if indeed we need to retain or replenish Krebs cycle metabolites, glutaminase might be a key player here, and there is a glutaminase inhibitor that is in clinical trials from several types of cancer. I'm not aware of a clinical trial with succinate dehydrogenase or even a preclinical study with succinate dehydrogenase, but the company who made this inhibitor called Calithera are certainly aware of that potential. Certainly I heard them discussing it in some meetings. But again, I'm not sure where it stands in the clinic right now. So, as you can see, there are thoughts in the field. There are some more advanced approaches. Some are already in preclinical or even in clinical studies. And maybe I should mention here that also there is now a HIF inhibitor, a hypoxia-inducible factor inhibitor, that is in a clinical trial for renal cell cancer. And there again, that might be, especially for partner glioma, I would argue, based on the genetics, but potentially for other SDH-deficient tumors, HIF-2 alpha inhibitor might be an important strategy to try and target in an organized clinical trial, succinate dehydrogenase deficient tumors. So this is where we are until today, and I hope I've been able to explain a little bit more about what is SDH and what can
Questions that were submitted, and I will start with the first question. What research is being done currently on SDHC epimutant GIST, and what is most current information known about that type of SDH-deficient GIST?
Yeah. Okay, thanks for that. And thanks also for those who submitted questions in advance, so it enabled me to prepare. So that's good. So yes, SDHC epimutant is it's relatively unusual way of shutting down a gene. I mean, yes, epigenetics and methylation is an important process in general that can shut down genes, but to lead to a direct link to a causal link to cancer formation is quite unusual. But in the case of SDHC in GIST, it was shown to be the case. So the studies that are ongoing, to my knowledge, are mostly to just characterize more cases like that or try to understand them genetically, not necessarily the consequences or the way to treat them. So as I mentioned in my seminar, treating SDH mutation can be relatively similar, whatever mutation or even epimutation it is, because what the consequences are in general is the loss of SDH activity in the tumor. But really, in the case of SDHC epimutant, one can imagine that if we can remove the methyl group that shut down the expression of this gene, maybe we can restore SDH expression. And because there are no mutations or deletions in this gene, we will restore wild-type activity or complete normal activity of succinate dehydrogenase. So it's quite an interesting approach. One need to use demethylating agents. There are some in the clinic. They are used for many reasons in different tumors, such as azacitidine or decitabine, for instance. They are approved drug, and they potentially can be explored therapeutically in these tumors. I'd like to add also that, as I indicated, SDH mutations in general will cause a hypermethylation phenotype, because they will, the succinate accumulated in these cells will inhibit the demethylases, the enzymes that remove methyl groups. So potentially using those drugs might be useful for any type of SDH deficient tumors. I'm not aware of any clinical trial related to that. Maybe you are. I am aware of a study published a couple of years ago that indeed demonstrated that the hypermethylation phenotype of SDH deficient cells is in correlation to sensitivity to the treatment with decitabine. So there is a rationale here, and there is a potential room for therapeutic intervention.
Thank you. And I just want to add, there is a clinical trial initiated by the NIH for guadocytabine that probably many people on the seminar know about, and hopefully will get expanded as well for this population. Okay, next question. Have you observed SDH families having non-tumor symptoms that could be related to the SDH major energy metabolic crisis? For instance, brain fog, headache, fatigue, exercise intolerance, or others. And can you describe if hearing loss is associated with SDH families?
Yes. Thanks for that. Yes, so here I'd like also to indicate that I don't see families. I'm not a geneticist or clinical geneticist or a clinician at all. So I'm not engaged with patients. However, I am familiar with the disease and many consequences of the loss of succinate dehydrogenase. And what I'm not sure here is whether this question was related to the so-called biallelic loss of succinate dehydrogenase. This is another disease where a patient received two mutated copies of one of the SDH gene from both parents. And in this case, the whole body, all the cells in the body are from birth are experiencing lack of effective SDH activity. This has a major metabolic consequence on the organism, on the human, on the patient in that case. Which include exercise intolerance, fatigue, and to some extent even hearing loss. But this is not, to my knowledge, I'm not familiar with a case where only when one bad mutated allele or copy of the gene is inherited in the germline, then all the body in these patients have one normal copy and they are fully functional. It's only in the tumor that the cells lose the second allele, the second copy of the gene. Under these conditions, I'm not familiar with any general metabolic crisis of the patients. So that more or less what I can characterize for this particular
Thank you.
question. Yeah.
Thank you. Okay, is SDH-D cancer, and are bilateral carotid body tumors, like paragangliomas, cancer?
Okay. Okay, so I hope I explained reasonably okay-ish that SDH-D is a gene, okay, is a name of a gene, and the gene product, which is the protein. Cancer is a growth of tumor, of cells that form a tumor. Cancer is a tumor that is grow uncontrollably, and also it's spreading and invading normal tissues. Not all tumors, however, are cancer. Okay, so we have benign tumors. Okay, so these are growth. They are in some paraganglioma or many paragangliomas, including carotid body paragangliomas, are not malignant. They are not cancer. It doesn't mean that they are not without any effect. They can have devastating effect, changes in blood pressure, headaches, effect of adrenaline levels in the case of pheochromocytoma. So even benign tumors can be very problematic and can also cause death in some cases. So need to be treated as well. But the definition of a cancer is a malignant process, which means that it's a growth that is also invading and spreading into normal tissues. If it's a local growth, then it's a benign growth, and then it's not considered cancer. Okay, and SDH is a tumor suppressor that its loss may lead to cancer.
Okay, thank you. The next question we've been asked a lot, and it's to really understand or explain the differences between SDH genes, like SDH-A, B, C, D, and what makes them so different.
Yes. So I try to address that a little bit during my talk, and I try to actually allude to the fact that theoretically there shouldn't be a difference, okay? Because if the mechanism of action, of tumorigenic action, is the accumulation of succinate, this happens due to the loss of SDH activity. And because SDH is dependent on every subunit, A, B, C, or D, the loss of activity of any of them will lead to the loss of function of SDH complex. So why do we see changes sometimes? Why do we see what you call phenotypic changes related to the genetics? Some are to do with mutations that may be not completely loss of function. They kept some activity. Some even generate some new activity, like making more redox stress, or as it's called, an oxidative stress in the cells. I'm speculating here. I just want to clarify that. The other reason to suggest differences between the different subunits is their location on the chromosome. So while typically, especially in germline mutations, there is a point mutation in the gene that affect only that gene, the second hit, which is the elimination of the normal copy, which occurs in the tumor, is typically associated with the deletion of a larger chunk of DNA around this gene. Now, around this gene, there might be other genes, or there are other genes as well. But of course, near SDHA, there are some genes, and near SDHB, there are other genes, etc., etc. So it might be that the deletion of some chromosome areas of SDHA, B, C, or D will have an additional effect that may dictate the severity of the tumors or other phenotypes that would be observed with the loss of succinate dehydrogenase.
Okay, thank you. How common are familial cancers associated with inherited SDH mutations?
So, in general, well, first, I don't know the statistics. I'm sorry about that, but it's very uncommon. Okay, so in general, familial syndromes are considered to be rare cases, and of these, SDH is certainly not the major one. Okay, I mean, I can name BRCA, for instance, in the case of breast cancer and ovarian cancer. It's being maybe a more familiar predisposition to syndrome to cancer. So the answer is it's very uncommon.
Okay. When someone is born with a germline SDHB mutation, do you think there's a second mutation involved for tumors to grow, whether epigenic or another faulty gene?
Oh, that's an easy one. Absolutely, yes. Well, first and above all, in most, if not all cases, there is a second hit on the same gene, on the second copy of the gene. So if SDHB mutation is a germline mutation, then the second allele of SDHB will be typically deleted. So that's a second hit. But even that is probably not enough. There is a need to accumulate more and more mutations. If we go, I mean, I won't go, but if we remind yourself on the oncoplot, this genetic study, you can see that for every patient there are multiple genetic alterations that are in addition to the SDH loss, the germline loss of SDHB. You need also some mutations. These are somatic mutations that are associated with oncogene activation.
Okay. Do you think supplements such as ubiquinol, methylfolate, vitamin C, riboflavin could help someone with a broken Krebs cycle?
Yes. Okay, here I need to, I mean, I'm talking from a rational scientific reason only. I'm not a great believer in such an approach. I don't see a real potential benefit here in most cases. It's going to be very difficult to fix a broken Krebs cycle with supplements. I mean, it's going to be impossible. So adding quinol or other supplements that theoretically can contribute to the respiratory chain are not going to help here. Again, from my understanding of the science. Of these mentioned, vitamin C may, interestingly, there is some potential rationale in using it because those enzymes that I mentioned, the dioxygenases that are inhibited by succinate that is accumulated in those tumor cells, they also, in most cases, require vitamin C as a cofactor. So one can imagine that maybe providing more vitamin C will enable them to function better even in the presence of high amount of succinate. Theoretically, it is possible. In reality, however, again, vitamins are so-called small. They are not in high concentrations in our cells. They are not required to be in high concentrations for a very simple reason. Their activity is reaching its maximal support even at low amount. We only know about vitamin from deficiencies rather than from over activity of these vitamins. So my suspicion is that even if we will take a lot of vitamin C, the dioxygenases will not work necessarily better. But that's now, at least I can see a link there. So of those mentioned, vitamin C may have some effect. But again, I'm not a supporter of such an approach in general. Okay, I'm sorry to say that. Yeah.
I'm going to try and fit one more question in, and I think a lot of the families on here are curious about different agents that could curtail tumor growth. So there was a number of different agents that were listed. Maybe you just want to comment on a few of them. A demethylating agent, also a low dose of a demethylating agent might work. TKIs, Temodar, a HIF-2b inhibitor, a PARP1 inhibitor. So, seems to be that people are curious to hear your thoughts in terms of what agents could curtail that tumor growth.
Okay. Thanks. Thanks for that opportunity. Again, my opinion is purely scientific and with my scientific knowledge and rationale. I must say that all of these have some good, rationale behind them. For instance, I already mentioned the azacitidine, the decitabine, etc. Agents that we already, I think, mentioned before. There is a good, there is a reasonable rationale to try and use them. And as I said, there are publications that explore that in tissue cultures, and they did demonstrate oversensitivity to these agents, and these agents are available for clinical use. So I would say, why not? But it needs to be studied clinically very well. Tyrosine kinase inhibitor, TKIs, VEGF receptor is a tyrosine kinase, and there are inhibitors for that. It is in use for SDH-deficient patients because the hyper-angiogenic effect, the hypervascularization, that is partly due to the activation of HIF. So yes, absolutely. I think it has been studied. At least I know about the study in renal cancer, and I'm not sure about GIST, but I suspect it is. Alkylating agents such as temozolomide. It's again, there is a potential rationale there. First, it generates DNA damage, and hypermethylated DNA is potentially more refractory to DNA repair. So it's more difficult for the cells to fix the damage. Interestingly, also, temozolomide or Temodar is commonly used for brain tumors, for glioblastomas. And over there, one indicator for its sensitivity is the level of an enzyme called MGMT. Not going into the level, but MGMT really repaired the damage that is induced by the drug. Okay. So when you have high MGMT, you are unlikely to respond to the drug, to Temodar. However, MGMT in some tumors is methylated itself and therefore is not expressed well. And these tumors might be more sensitive to Temodar. So in this case, considering the hypermethylation profile of SDH-deficient tumors, one will need to look at MGM2 expression level, and if indeed it's low, then there is a rationale in trying to use temozolomide. But again, I don't know enough about its what is called pharmacokinetic or pharmacodynamic criteria, how well it gets into the tumors that are relevant. I know it's relevant for glioblastomas. I'm not sure about other tumors. And also there are side effects, of course, associated with this treatment, severe side effects as well. So it's less of a targeted therapy. It's more a generic DNA damaging agent. HIF2, we already mentioned. Certainly, I think there is a good reason to use HIF2. But again, from a genetic supportive role, I think paragangliomas with SDH mutations are more likely to benefit from HIF2 inhibitor. I don't know much about the role of HIF in GIST, for instance, an SDH-deficient GIST. I haven't seen any evidence for that. And then the PARP inhibitor, in a similar way to the alkylating agent, it does generate DNA breaks due to inhibition of processes that are required for DNA repair. It is in clinical use, particularly in BRCA patients that I mentioned before. But there was a recent study in Nature Genetics that demonstrated that SDH-deficient cells, due to the hypermethylation process, are more sensitive to PARP inhibition. I think, if I remember correctly, that it is already in a clinical trial for SDH-deficient tumors. I'm not sure which. So yes, there are many ways beyond the direct metabolism, etc., and all of them should be explored. Some are more rational than others, but unfortunately we don't always understand everything, and thinking only rationally, there's always good reason to exclude things from a rational point of view, but seriously, I think if there is some mechanistic potential to support, I would definitely try and explore this clinically.
Great.
Sorry, it was
Thank
a bit
you
too long,
for answering.
maybe the answer.
Thank you so much for answering all these questions. Unfortunately, we did not have time to get to all of them, but thankfully, Dr. Gottlieb has compiled answers to these questions that were submitted, and we're happy to distribute that to participants after this event. So at this point, I would like to thank Dr. Gottlieb for joining us today and presenting on this very important, relevant topic, and volunteering his time to speak with us in his evening, as it is now evening time over there.
Yep.
And if anybody has any further questions, feel free to email me, Sarah Rothschild, and I'd be happy to send that over to Dr. Gottlieb as well. So thank you very much to everybody who joined us today, and have a wonderful day or evening. Enjoy.
Thank you.
Bye.
Bye-bye.